Hi everyone, my name is Zeyuan Chen. I'm a PhD student in around Halperin's lab at UCLA. Today, I'm going to present our latest work on a unified model for cell type resolution genomics from heterogeneous omics data. We now have a very rich depository of bulk epigenetic data collected from complex tissues such as blood, adipose, and brain. We denote those bulk profiles as matrix X here. At the same time, we're also aware that there's some inherent heterogeneity in X because they're actually mixtures of several distinct cell types. So we visualize it here as cells with different colors. There are several attempts that tries to disentangle the signal computationally. One earlier approach is try to treat this as a matrix decomposition question, where we're trying to decompose bulk profile X into two matrices, W and Z. W is what we call the cell type proportion matrix that is sampled by cell types. Each row denotes a specific samples, and each column denotes a specific cell type. So for example here, the three circles indicate sample 1's yellow cell type proportions, green cell type proportions, and blue cell type proportions. The cell type specific profile Z is assumed to be shared across population, and each row denotes a specific cell type's profile. We can then model our bulk level profile as a weighted average of the cell type specific profile, and the weights are precisely the cell type proportions that are sample specific. There are several methods in this decomposition framework, notably the cybersort for RNA and epidish for DNA methylation. In this talk, however, we're going to try to tackle a slightly more challenging question, which we call the deconvolution question. Similar as before, we also have our cell type proportion matrix W, but this time we're trying to estimate a 3D tensor that is cell type by feature by samples. So now the key difference is that the cell type specific profiles are also sample specific. We also model the bulk level profile as a weighted average. The weights are still coming from the sample specific cell type proportions, but now the Z have three indices. Some of the methods that falls under this framework are cybersort X for RNA and TCA for DNA methylation. We can better understand what is the key idea of our newly proposed framework by just focusing on one specific feature, J, and this time we analyze what is the covariance structure between the cell types. Pictorially, we only listed three cell types. So the variance covariance matrix is going to be 3 by 3, but more generally, it's going to be a K by K matrix. We can also actually understand better what's the difference between methods by trying to see what structure is it trying to capture. For example, for cybersort X, it assumes this is a diagonal covariance structure, and it also allows certain variance term, for example, the one in the middle, to be set to 0 if heuristically it thinks there's not enough variation in that cell type. For TCA, it also assumes a diagonal structure. It, in addition, also puts some distribution assumptions, in this case Gaussian distribution, on top of the data. For our newly proposed framework, there's two key innovations. The first one is that we're actually modeling the entire full variance covariance matrix. And the second one is that we're not restricting this to any specific distribution, so it should be applicable to any distribution and thus any omic type. And all of these are partially inspired from single cell technology. So what's shown here on the left panel is the UMAP from a PBMC dataset. What we observed is that the cell type from the same lineage tends to be more correlated. For example, the CD4, CD8, the natural killer cells that are from the lymphoid lineage are more correlated within this group than if we were to compare this to the blue cluster, which is the monocyte from the myeloid lineage. So we wanted to come up with a metric that quantifies how strong is this co-expression pattern amongst the cell type, or how strong is this off-diagonal signal in a variance covariance matrix. So the metric we end up using is the Van Neumann entropy. Note that this is actually not the same entropy as what people typically use in the cross entropy laws. This entropy is calculated on top of the k by k correlation matrix. It takes value between 0 and some number, but a specific upper bound is related to the dimension of the matrix, so it could be larger than 1. The key takeaway of this metric is that usually smaller number indicates that there's very strong co-expression pattern amongst the cell type and relatively strong off-diagonal numbers in their correlation matrix. On the other hand, a very large entropy indicates that each cell type is behaving almost independently, and then there's almost no structure off-diagonal. So what we're showing here on the right panel is the histogram of the entropy calculated per gene across all genes in this PBMC dataset. So we want to point the audience to the fact that there are certain non-trivial amount of genes that are harboring this very strong co-expression pattern amongst the cell types, so it justifies our attempt to model the entire variance covariance matrix. With that in mind, let's dive a little bit deeper into the full model specification. We call our model TCAX. So as before, it's easier if we're just focusing on one specific feature, J. And over here, we're saying the cell type specific profile has this expectation, mu J, that's going to be number of cell type by one vector, so a k by one vector. And as alluded before, the covariance structures of this cell type specific profile is sigma j. So we're actually modeling the entire k by k matrix. The bulk profile is a linear combination of the cell type proportions times the cell type specific signal plus some IID noise. So we're going to set the core parameters of that feature j to be the mean, mu j, and then the covariance, sigma j. The model itself is capable of handling cell type specific covariates and some technical covariates. We're not listing them here just yet for simplicity reasons. Another thing you might note is that we're not listing W, the cell type proportions, as one of the model parameters. And indeed, we require some external tool to give us this initial estimation of cell type proportions. So this is going to introduce additional unaccounted variation in models, but as we're going to show later, all of our real data analysis is carried out using estimated Ws. And also prior literatures have well established, especially those benchmarking paper, that current method is very capable of capturing this cell type proportion information very reliably. We're going to briefly talk about the actual model parameter estimation step in the next slides, but once we have those estimated data, we can start our inference. Now we're going to set our TCAX best estimation of this cell type specific signal to be the conditional expectation of Zij, conditioning on the learned model parameters, the observed bulk level profile, and the estimated cell type proportion. And it turns out that it has a very nice closed form solution. You can conceptually break this into three major pieces. The first component is asking what is the expected cell type level profile. And the third component is asking what is the difference or what is the deviation away from the bulk level profile. And the middle term acts as like a coefficient for distributing this amount of deviation to each of the cell types. And very briefly, without getting into the technical aspect of the actual optimization scheme, we're just going to say that it is inspired from the generalized method of moment. And in practice, we do iterative update. Within every iteration, we're also going to update sample-specific weighting scheme. Another trick we use is to reparameterize the covariance matrix with the Chomsky decomposition. So instead of directly optimizing over the sigma j, we're going to optimize over Lj such that when we're actually reconstructing back the sigma j, it's guaranteed to be positive semi-definite. This weighting scheme does achieve asymptotic consistency, indicating that as sample size goes to infinity, we're expected that the estimated parameters converge in probability to the true model parameter. We show our first piece of evidence via simulation. So we went back to the PBMC single-cell RNA dataset, and we constructed our cell type level pseudobulk and used that as our ground truth tensor Z. We then counted how many cells does each sample have at each cell type, and that gives the ground truth W, cell type proportions. And with Z and W, we're going to construct our heterogeneous pseudobulk X. Next, we're going to give this W and X to 4 competing methods and ask them to do the parameter estimation and tensor estimation, and eventually give us the estimated cell type specific profile Z hat. We then will compare gene by gene and cell type by cell type what's the correlation between the true versus the estimated. You have already seen CyberSort X, TCA, and TCX. We added another baseline model to the mix. This is the easiest way we can come up with that give us cell type specific profile directly from bulk, which is just distributing that bulk profile to each cell type by their sample specific cell type proportion. In the bottom, you see 4 different panels. Each panel represents one specific cell type, and within a panel, it's broken down into high entropy basket and low entropy baskets. The panel themselves are ordered by cell type abundance. So the CD4 T cell is the most abundant one, followed by the natural killer, and the B cell, monocytes, is the least abundant. And what we're showing here are the box plots calculating the correlation between the true and the estimated tensor for each gene calculated across all samples. Since TCAX actively models the 4 variance covariance structures, you would expect that it benefits more in the low entropy baskets. And indeed, that's what we observe. TCAX is significantly better than all the competing methods in all cell types actually, but especially benefits in the low entropy group in the less abundant cell type. So intuitively, you can think of it as it's borrowing information from the other cell types to help its prediction with the less abundant ones. We did one additional test, which is to check if TCAX estimated tensor captures more signal in the true tensor than bulk profile does. So we fit the true tensor against the cell type proportion W, the bulk profile Xij, and the estimated tensor Z hat IJH. Ideally, we should see stronger statistical evidence of significance on the leading coefficient alpha 3 in front of the estimated tensor than on alpha 2, the leading coefficient, in front of the bulk profile. We fit this model separately for each gene, and the results are as follows. It is also broken by cell type. Within each panel, each dot represents a gene. X-axis is plotting the negative log p-value on the leading coefficients alpha 2. Y-axis is plotting the negative log p-value on the leading coefficient alpha 3. Very surprisingly, TCAX is the only method that can cross the diagonal line in all cell types, indicating there's a stronger significance. We next switched our attention to methylation data. Over here, we have a small data set that has the bulk level methylation profile, as well as cell type specific methylation profile collected from purified blood. Unfortunately, this dataset is very small, only 6 samples, so it's unrealistic to expect any model can learn any parameter meaningfully out of 6 samples. So instead, we went to a larger dataset that has over 426 samples. We asked some decomposition method to give us an estimate of the cell type proportion W hat, and then we're going to use the bulk profile as well as the estimated proportion to learn the model parameters for TCAX in this larger dataset. We similarly use some decomposition method to get the cell type proportions for the smaller dataset, and with that, we're just going to transfer this data, learn in the larger dataset, and use that to estimate the tensor for the smaller ones. Similar as before, we wanted to compare the cell type level correlation for each CPGs between the true and the estimated tensor. However, there's only 6 samples, so correlation calculated will be very unreliable. And instead, we're just going to do the following. For each cell type, we're just concatenating all CPGs after adjusting the mean methylation value, and then calculate the correlation. So it's going to be one number per cell type. We did this extra step of adjusting the mean to enforce that the performance is not driven by just predicting a general trend of the mean methylation fraction well, but it has to get the sample-specific signal right as well. If we focus on the left panels, rows represents different cell types. They're also ranked by abundance. The first row represents granulocytes, which is the most abundant one, and the last row represents B cells that are only 6%. Columns represents different methods, and within each panel, x-axis represents the cell type specific profile after adjusting the mean. The y-axis represents the estimated tensor after adjusting the mean. Each dot is a CPG site. The expected behavior is that the closer you are along the diagonal line, the better or the more concordant you are with the ground truth. And you can already see that TCAX shows a much stronger improvements over previous method, especially in the less abundant cell types. We also analyze our results by broken this into 2 baskets, the low entropy and high entropy ones. On the right hand side, each panel represents 1 basket. The x-axis are the cell types sorted by abundance, and the y-axis are the correlation between true and estimated tensor. Similar to what we've seen in RNA, in this real data methylation analysis, we see there's a huge improvements in the low entropy baskets because TCAX is indeed modeling those covariance structures actively. Another thing to note here is that CyberSort-X almost completely failed other than in the most dominant cell types. It's not surprising because this is originally designed for expression data. So we just further excluded this method from any real data applications on methylation. We then checked some downstream application of deconvolution methods. The first one we checked is cell type specific differential expression in follicular lymphoma tumors. So a quick intro, follicular lymphoma is actually the second most indolent non-Hodgkin lymphoma in US. It's accounting for nearly 20% of the cases. There are prior studies using FACS-sorted B cells from FL tumor and has identified over 600 differentially expressed genes between conditions. And the conditions are with or without a specific CREBBP mutation. We are curious to see if deconvolution methods can capture this signal that are previously only found via effect sorting. On the left figure, we already broken the differential expressed gene sets to 2 major components, the one that are mutation downregulated and those that are upregulated. The y-axis is plotting the log2 differential expression signal and the estimated B cell sample-specific expression profile. Each dot represents a known DE gene reported by the original study. You should expect the majority of the dots fall below the y=0 line in a downregulated set and over that line in the upregulated set. And TCAX is the only method that does better than a straightforward analysis on bulk, as indicated by this orange and brown color. It is also statistically significant better than every other method with a paired t-test, except in the upregulated group of the genes, it might appear that the baseline model is somehow doing the best. It turns out that it is due to how we construct baseline. If you look at the right-hand side, you'll see that the estimated cell type proportion of B cell is larger in the mutated group compared with the wild type. And the baseline model will artificially distribute a larger fraction of the bulk expression profile to the mutated group in the B cell. So it's going to do this regardless of the true direction of the underlying differential expression pattern. And indeed, it backfired because if you look at the downregulated gene sets, majority of those are still predicted to be upregulated by the baseline model. And finally, we're going to apply our deconvolution framework to epigenome-wide association study. So EWAS is similar to GWAS, where we have a phenotype of interest Y, and we usually just scan through the genome, CpG by CpG, and check if there is a significant association between this phenotype and the CpG methylation profile. Traditional EWAS has been carried out under heterogeneous bulk profile, and now with the deconvolution method, we're asking a higher resolution question. We want more than just the CpG that are associated with the phenotype of interest. We also want to pinpoint to which cell type is it associated with. We can very naturally achieve that under the current TCAX framework by just adding one additional extra term. The expectation of cell type specific profile at sample I, CpGJ, can be written as mu j plus an additional phenotype of interest times its effect size. And note that this effect size is also a vector that is k by 1. Similarly, we have the covariance structures as before, and the bulk level is modeled as a linear combination plus some IID noise. Checking for cell type specific EWAS hits then reduces to check for entries in gamma j that shows strong statistical evidence of non-zero effect size. For example, here, if the first entry shows strong evidence, then we can pin this association to the first cell type. You'll soon see that different assumptions on the covariance structures will be reflected in the final consistency performance of the cell type specific EWAS hits. So coming to the result, let's first focus on the right panel. So here we're listing 3 methods. We exclude the CyberSort X and included a new method called CellDMC. Essentially, it fits a regression model on the interaction terms between phenotype of interest and the estimated cell type proportions. Mathematically, it can be shown that this is equivalent to assuming a diagonal covariance structures with all of the cell types forced to have the exact same variance. And on the left panels, we have on the rows the 2 major phenotypes we investigated, sex and age, and on the columns, 3 different methods we investigated. Since we don't know the ground truth cell type specific EWAS hits, we report a consistency score here. Intuitively, this metric tells you how well can you expect your EWAS hits to be replicated in an independent data set. For each figure, we first run EWAS on the discovery dataset listed in the rows. We then go into an independent different dataset listed in the columns and try to validate the result we got from the discovery dataset and report the F1 score. Higher F1 score indicates better consistency, and as you can see, through all pairwise comparisons between 4 datasets, TCAX achieves the best mean F1 score in both age and sex. So to wrap up this presentation, the key takeaway message is that we're proposing a new distribution-free deconvolution method. The method is capable of handling full covariance structures between cell types, and because we kept it distribution-free, it should be applicable to heterogeneous bulk genomic data of any type. Empirically, we see improved deconvolution results in both single-cell RNA in-silico mixtures and real-world DNA methylation data. It also shows superior downstream task performance, for example, in deconvolution of tumor RNA and identification of cell type specific EWAS hits in methylation data. Finally, I'm going to thank Elior Rahmani, who contributed as much to this project as I did, and also my advisor, Eran Halperin. A beta version of the code is available upon request, but we're in the middle of formally packaging this and deposit this. All right, thanks for your attention. I'll take any questions.